thanks for the invitation to speak today, and thanks to SoilsWest. I'm happy to be joining SoilsWest. Been here 3 years, and it's about time. So, my talk today is going to be on soil spectroscopy, I've chosen a really, fantastic title: "A Technology for the 21st Century." I actually really believe it, and I hope that by the end of this talk, you'll also believe it. The application of spectroscopy is still very much in the research phase, and I think it should be applied. It's, at least some aspects of the method should be in the practical space. And the reason it is not in the practical space is because I think that there's been a few false starts in the past. We've been working on spectroscopy for, I don't know, 40 years, 30, a long time. But there's been lots of false starts because I think people have been a little bit over enthusiastic about it. I don't think there's been anything, sinister or anything. I think people have been over enthusiastic and they've sort of oversold the technique a bit too early. And often because they thought they were going to make some money. But I think that now we know more. Right? We know more, and we're not going to make the same mistakes, because I hope that I can show you that the technique is actually ready. At least some aspects of the technology are ready for us to be using it, because we actually need soil technique, and this is a really good way to do it. So my presentation today is a little bit of show and tell. I've got lots of slides, nothing that I will really delve deeply into, lots of pictures, but I hope you get the point. So just to give you a little bit of a summary of what I'll be talking about, I'll give you a little bit of context, like most of my lectures. I often start with a little bit of context. Then I'll tell you about spectroscopy, or my view of spectroscopy, some fundamental stuff. You can probably find the textbooks, but okay, I think some of you might be new to the method. And then I will show you a few applications, right? And a lot of this work I've done in the past. There's not a lot of new stuff here. But the applications show you the potential of the method, and then I'll talk about digital soil mapping a little bit. I'll talk about carbon modeling a little bit, and then I'll give you some recommendations, even if you haven't asked for them. I will also give you some final remarks, and then I'll finish. Let's start. I've got notes because otherwise I'd ramble, and my 30 slides end up being 2 hours. I think people cut me off. So anyway, currently, as everybody knows, populations are increasing. We're going to have 11 billion by 2100, and so this is putting pressures on the world. We're consuming more and more resources, and we're producing more and more wastes. And of course, this is not only affecting us, but it's also affecting the planet entirely, right? So soil is central to all of those existential challenges. Soils are these natural integrators, and it's nicely summarized in this picture by Lau. It is really where the cycles of matter and the transfers of energy all interact, and it's central to the interactions with the atmosphere, the hydrosphere, the biosphere, the lithosphere. And so it's obvious then that soils are used to produce a lot of the ecosystem functions that we rely on. So soils produce all of these functions, but I'm not going to read all of them. I'm sure you all know them, and it's for that reason that soils are really central in the political agenda at the moment. The soil is present in, I don't know, 7 of the Sustainable Development Goals. It is central to the UNCCD and UNFCCC, and the FAO has a lot of programs on soil through the Global Soil Partnership. So I think that cost-effective measurement is crucial for us to develop solutions to address those existential challenges. And I like this quote by Lord Kelvin, but I'm not going to repeat it because I think it's on my SoilsWest profile as well. But really, without quantitative soil measurements, science kind of fails, right? And also, we can't really assess soil. We can't monitor baselines. We can't see how soil reacts to certain interventions. And so measurement is critical. And of course, there are different types of sensing technologies that we can use now to measure soil. And I've sort of categorized them in this electromagnetic energy. We have the X-rays on the left, radio waves on the right, and there are a lot of small, affordable sensing methods that can be used to measure soil properties. You have gamma radiometers that measure mineralogy. You have X-ray sensors that measure elemental composition, mineralogy, all sorts of things. On the other side, you have electrodes, miniaturized electrodes, ion-sensitive field effect transistors that can measure nutrients. So really, the technologies have developed so much over the last 10 years that I think that there are lots of opportunities for us to use sensing, not to replace conventional analytical methods, but to work together with the conventional methods, because a lot of these sensors rely on calibrations to those analytical measures. So a lot of people think that people working on sensing are trying to replace what people currently do. It's not the case. So at this point, soil spectroscopy is probably the most advanced soil sensing method that there is, largely because we've been working on it for 30 years or so. So what I will do then is I'll tell you a little bit about soil spectroscopy. So what is it? It is the interaction of electromagnetic radiation at specific frequencies with the soil. And it's these fundamental physical processes that enables us to measure the composition of soil. The most common frequencies that people use are the mid-infrared, MIR, the near-infrared, NIR, and the visible, but there are, of course, other ranges. There's the UV range that people actually use to measure some elements, nutrients, and the far infrared, which people use to measure mineralogy primarily. So what happens when the light, mid-infrared or near-infrared light, shines onto the soil? The light scatters in the sample, and it causes the bonds in the molecules present in the sample to vibrate. And these vibrations can be different. You can have bending vibrations, you can have rocking vibrations, all kinds of vibrations. But some of these bonds then absorb the energy, and only some of it is reflected back. And that light is diffusely reflected back. And then the detector captures that signal, then you get the spectrum. So in the mid-infrared, you get these fundamental vibrations. What this really means is that when the light hits those molecules, they jump from a ground state to the first energy state. And so they're called fundamentals. And those bonds, as I said, vibrate differently. In the near-infrared light hits those molecules, they jump from a ground energy state to a second or higher order energy state. And those are the overtimes. Now, those jumps can happen between 2 or 3 or different energy states, and then you get these combination bands, which make the near-infrared spectrum actually quite difficult to interpret. So in the visible range, which is an important range, I really like the visible range. Some people ignore it, but I think what we can see is like so useful. It's a totally different process. So these are electronic transitions to electrons in different electron shells. So these are some examples of soil spectra. So you have the infrared on the right and the infrared on the left. Now, because of the processes that I described, the mid-infrared spectra look a lot more interesting. There's a lot more peaks and troughs in the spectra. While the near-infrared is rather dull, right? There's only a few absorptions, but don't be deceived by its appearance. There's a hell of a lot of information in those lines on the right there. It just means that you perhaps need to be a little bit cleverer, or it's a little bit trickier to extract the information out of it. And so then you need to rely on multivariate statistics or machine learning these days to try and extract the information from that spectral range. Now, the use of different spectral ranges depends on various factors, and what range to use really depends on what you want to do. Each has advantages and disadvantages. So, for example, the mid-infrared technique requires a significant amount of sample preparation. So if you're prepared to fine-grind the sample, dry them really carefully, fine-grind the samples, they can afford to do that, mid-infrared might be a more suitable technique for you because obviously it contains a hell of a lot more information. However, if you want to use the techniques in the field because you want to get some rapid estimates on some properties, some rough ones, just to tell you where the properties might be high or low, then you buy yourself a near-infrared spectrometer and take it with you to the field. You can't do that with them. So it depends what you want to do. So in terms of what information is in this spectra. Now you remember your basic soil science, what's soil made of? Water, air, minerals, and a tiny bit of organic matter. Well, the spectra measure these things, all of these things. So a single spectrum can tell you about the iron oxides that are present in the soil, the things that give the soil color, tell you about clay minerals, things that give you soil cation exchange capacity and so on, nutrient holding capacity. Tell you about if the soil is under field conditions, it will tell you how much water there is in it. And it will tell you if the soil has carbonate because there are specific absorptions that characterize carbonate. It will tell you the type of organic matter. And now this is a little bit tricky. There's a hell of a lot of research currently trying to untangle the organic matter signal from these soils. And the reason is that soils are really complex. So the organics that you get in the soil are kind of tricky, but it is possible, and there's quite a lot of research that is starting to demonstrate that this approach can characterize the biological diversity of organic matter. So I cannot even tell you about particle size. So you can tell a single spectrum can provide you information on soil. Most of its properties, and it's the reason why you can build these models to estimate soil properties. Right? It's not like magic or anything. It is based on a robust physical science. This approach. Now, just to prove to you, here are some spectra of some minerals. So you have near-infrared, mid-infrared, some organic matter, and some water. So you can see that, the spectra can measure the hematites and the goethites, as I said, those iron oxides, carbonates, kaolinite, illite, and smectite, those clay minerals. The near-infrared can't really measure cores, right? It's sensitive to cores. So you can see that the near-infrared or the mid-infrared can. Lots of different organic functional groups occur in the mid-infrared. I know you can't see that, but these are spectra of, for example, peat and compost and humic and fulvic acid. So you can see that they actually have response in those regions. And on the right, we see spectra, near-infrared spectra of soil water, and you can see that the response of soil water in that range is quite linear, actually. So you can estimate, or you can measure fairly directly water content with this. Now, for all those reasons, and more, it's not surprising then that there's been a hell of a lot of research on this technology. And the number of papers that have been published has increased exponentially since I left uni in the 90s. So funny, I was talking to a student the other day, and I was telling her about how old my bike was. My bike was a 1978 bike. She wasn't born till 1995 or something. I was going, what? When did that happen? Anyway, so much of this research is focused on the modeling component of soil spectroscopy. There's a hell of a lot more that people haven't done. And it's not all about modeling. I love modeling. I'm a modeler. It's not all about modeling. There's a hell of a lot more that we can do with the spectrum. Some other reasons why people like these technologies. One of them is that you can measure this spectra from different platforms. So you can measure it from satellites. More and more, you're getting these hyperspectral, high spatial spectral resolution sensors that can measure in the visible and near infrared. You can measure it in the lab, of course. You can measure it in the field, either statically, you get your probe and you put it on your soil profile, or you can even put it on the back of an instrument. And there are some commercial systems that have got these spectrometers on tines and things, and they get dragged along the field with them. So the technique is really highly versatile. And there's quite a lot of research right now actually trying to fuse and combine these different the measurements from these different platforms, because you can see that there are advantages to each of those platforms, right? So the remote sensing cover lots of area, while the proximal sensors can actually do something that the remote sensing methods can't, measure down the profile. So the integration of those technologies is currently really, really, really important. Yet another reason is that the cost and the size of the spectrometers has dropped quite dramatically over the 20 years or something. Since I left TV. So, you know, what was affordable in the 90s was something as big as this desk, right? And you wouldn't be able to lift it yourself. You needed two people to move it from one place to another. So now, where you can have something that is as big as your remote control or even smaller, linked to your app, so you can actually measure things. Now, I've got a note on the bottom there saying that this accessibility to these technologies is actually really good. I really like it, but actually, it's not all positive, because unfortunately, it's given some misinformed entrepreneurs the idea that you can simply just go and buy one of these things and then go and measure, and all by magic, you'll be able to make lots of money measuring carbon. It's not that easy. And the problem is that we've had this before, you see. I mentioned that in the past we had these false starts. I don't want that to happen. Okay. So just a little bit of research. So we recently looked at all of these miniaturized spectrometers. So how well do these little things perform? And they are little, right? So this thing is probably a little cube this big. This thing, well, you can see how big that one is, right? This thing is an OEM thing, so you can actually build your own case and do what you want with it. That is the most produced one, I guess. It's a German company who have also attached it with an app, so they give you the spectrometer and an app, so you can actually collect and look at things on your phone and do crazy things on your phone and put it on a cloud. So what we did, we compared those. So we got them all, had some really nice samples from a mine site where they were trying to assess soil health. So they were measuring all these different indicators, and they used these indicators using some crazy magic to derive a soil health assessment. So we said, oh, perfect data set to sort of do this comparison. And so we measured everything with the little sensors, but also with this sensor called E down there, which is sort of like the benchmark. It's what most people use to do near-infrared spectroscopy in soil. And what we found was rather amazing, right? So if you compare it, so this is the concordance correlation, right? That counts for bias and imprecisions. It's a really nice correlation. The higher, the better, of course. But what you're comparing, and I hope you can see it, is the bars. So the different colors are for the different little sensors, and the pink one is for the big one, right? But you see there's not much difference. And in fact, for some properties, you see the little ones in combination was actually better. Right? These are reduced spectral ranging. So this is only a visible sensor. This only does 1700 to 2000. This is 1300 to 2500, and that's similar. So we combined some of these sensors to make more complete spectral range, and they worked. Right? And just as well. But the difference is that this sensor, this big one, costs between $70,000 and $100,000. Well, the most expensive of these little sensors was $10,000, and the cheapest was $4,000. Okay, so how do you do a spectroscopy? So if you want to do any sort of spectroscopy, you really need to develop a spectral library. So it's whether you want to measure things directly or you want to do some modeling. And this diagram is meant to summarize that a little bit. So the first thing that you do is decide your domain, right? For example, this is Australia, but it could be your field, your farm, or whatever. And then you design a sampling strategy. I won't stress that enough. You need to design a sampling strategy. Don't just go and haphazardly go and collect samples willy-nilly. Design it. Go and take your samples and do the sampling right. Then you could take the soils and prepare them, if you're using near infrared, you probably don't have to do too much sample preparation, but if you're doing mid infrared, you need to find brightness. You prepare your samples. Then you take them for analysis. You do your chemical analysis, you measure them, those same samples with a spectrometer, you put those in a computer, you store them in your database, the spectral library, and then you're ready to go and do some modeling. And some people, and me included, have used archives as a source for soils to build these spectral libraries. And that's cool, that's fine. But you have to be aware that the models that you derive might not be necessarily applicable everywhere, because the soils in those archives were not sampled for building spectral libraries, were sampled for very different purposes. So you've got to be a little bit careful how you use spectral libraries when they're built from legacy data. A little bit about the modeling. So once you've built your library, put it in your computer, you've got the database, you go and do some modeling. This is a little bit of a summary of how things developed over the last, 40 years or whatever it is. So at the start, we tended to use multivariate statistics, things like principal component regression and partial square regression, which were developed at about that time in the 1980s by chemometricians, chemists really, that were really quantitative. And so we use those techniques for relating the soil properties with this multivariate spectrum. And these spectra are really multivariate, right? Because you get, for a near infrared spectrum, there are potentially 2,000 wavelengths. And those wavelengths are highly correlated. So you've got to treat those data, judiciously, I would say. But the mid-infrared spectra, there could be even more, right? So they are highly multivariate. And that's why those multivariate statistics with principal components and partial least squares regression were the ones that people sort of, adopted really quickly, because those methods are particularly useful for dealing with highly correlated data, multivariate data. In the 90s and 2000s, machine learning started becoming a little bit more popular. So those methods were adopted by us and used. And more of late, in the last, 5 years, these deep learning convolutional neural networks are the most popular methods to use for spectral modeling. I like all these methods, I really do. But sometimes I think that you don't really need a big sledgehammer to, you just need a little nutcracker. And sometimes, and that's what people do, and particularly people that are new to the spectroscopic method, but are really keen on data science. So they want to use the latest, best algorithm, but actually you don't need to all the time. So this is one, so there are a couple, so you build your library, you can model things fine, but there are some challenges to doing spectroscopy. So one of the misconceptions of many that are new to the field is that for soil spectroscopy to work, one needs to develop a large and diverse spectral library, and then you need to use some clever model, right? Whether it's machine learning or statistical, and then you can make predictions anywhere. It's a misconception. It doesn't work like that, unfortunately. It's a really cost-effective way to do things if it worked, But what we find is that the models that you build tend to be highly biased. Right? Because those models are not capturing the variation locally, there's too much muddling the models. And sometimes those models don't have the information that is at that local site. So what's the alternative then? There's been a lot of people writing that building spectral libraries is useless then, right? Because if you can't use them to do exactly that, build a model and then go and predict somewhere in Western Australia or in New South Wales or in China, what the hell is the use of building these global libraries? I don't agree with that, and I'll tell you why in a minute. But the other way is that then you need to build local models for every case. So if I want to predict carbon in Southwest and Western Australia, then I should build a library for Southwest and Western Australia. That's cool, that works, but it's expensive if you have to do that everywhere. Right? You get unbiased models, unbiased estimates, but it's expensive. So there must be other solutions. And the solution, there's lots of solutions tried, but the solution is try to localize these large spectral libraries so that they have some ability to predict locally. So there's been methods like spiking. Those of you who are into spectroscopy may have heard of those methods. Spiking, memory-based learning, but they don't really work consistently everywhere. So what we've been doing is we're looking at this method called transfer learning. It's not a new method. I thought it was a new method, but it's not a new method. It was the first paper of transfer learning in the 1960s, I think, but because of all this machine learning interest, it's sort of been brought back out. What transfer learning is, essentially, is you use knowledge that's stored in you. So like most machine learning things, it comes from some sort of human thing. So you use information that you've stored and you apply it to a current situation. So an analogy is that, when you learn to ride a bicycle, right? You learn to ride a bicycle, you store some information in you so that when you go and want to ride a motorcycle, you bring some of that back, some of the useful bits that tell you about balance and things, so that you use that information and so that then you can ride a motorcycle easily. And it's the same sort of idea with this transfer learning. So here with transfer learning, what we need is just a few samples from your local site so that we can actually combine that with the learned information from the library, and then we can model that. And it works. We just published the paper. I'm not going to go into any of that detail, but if you're interested, please go and read the paper. And we compared this. So we did a fairly robust assessment. So it wasn't just for one data set. We used a global spectral library with more than 50,000 spectra. We tested the method in the US on a corn farm in the US, on an oat field in Sweden, somewhere in Australia, and somewhere in China. And the method consistently worked well. So this doesn't mean that we don't need any lab analysis. It means that we'll minimize the number of samples that you need to collect at any site to make an accurate local model. The second challenge is Geiger concept. And it's a really important one. It's really relevant in soil spectroscopy because for two reasons. One, the method is highly reliant on empirical calibrations, right? And two, because of empirical calibrations to the analyte, so something that's measured in the lab. And in a calibration, it's reliant on modeling. So I think that the spectroscopy itself is not the problem, because spectroscopy is one of the most reproducible and repeatable methods, analytical methods that there is. That means that if you have a spectrometer and you have a soil sample and you measure it, right, and you go away and come back the next day and you measure that same soil sample, you take that soil sample to another lab, even if it's a different instrument, you measure it, you'll see that the variation is rather small. You will still get the same absorptions due to the minerals and organics that are present in the sample. problem is with the modeling. And the modeling relies on the analytical methods that you use to build those models. So you need to take care of the reference soil analysis. One of the things that I absolutely recommend if you're doing spectroscopy, never believe a lab. Doesn't matter if it's ASPEC accredited, NASA accredited. No, always put some blind duplicates in there, right? And send the blind duplicates. And don't tell them, of course, that you sent them that. Only tell them if they come back really different, which 80% of the time that I send samples, I have to call the lab. Doesn't matter who the lab is. Soil sample handling and preparation is really important. How you handle the soils, how you prepare it. Do you dry it at 40 degrees, 80 degrees? Do you grind it? Do you mill it? What's the particle size? All of these things will affect your spectrum. The modeling, right? It's not straightforward. I think spectral modeling is one of the toughest modeling fields that I'm aware of. I've done modeling, spatial modeling. I've worked with lots of different types of models, and spectral modeling is harder. It's trickier. It's not harder. I would say it's trickier. There's lots of holes that you can fall into. So you need to be careful. You need to study it. You need to learn how to model. You need to learn how to validate. Right? You need to learn to validate those models. And validation isn't as simple as just taking 30% of the data, putting it aside, building a model and validating on that. You need to think about how that data was sampled. Are those samples independent from your models? And if they're not, well, they should be. Right? So you can assess those things. And finally, I think, as I mentioned earlier, if you are developing a new spectral library, think about your sampling design. Right? Really, like most things in soil science, everything falls back to the sampling design. Okay, so that's the spectroscopy thing. I'm probably running way out of time. So just some applications. So as I mentioned earlier, this spectra can be used to directly quantify minerals, color, and things. So this is what we did a little while back now. So we measured samples from the archive, from the National Geochemical Survey of Australia, whatever we could get our hands on. We measured, there were about 5,000 of those, and then we analyzed the spectra, and we looked at bits that belong to, or that relate to smectite. It's a clay mineral, as you know, it's the one clay mineral, illite and kaolinite, right? And then we use this digital soil mapping or spatial modeling approach that relates those measures with environmental covariates. And we used some machine learning method to build that relationship, and then we extrapolated those models across the whole country. This was actually the first time somebody mapped such a big area, 90 by 90 meter pixels, so it's a football field size across the country. And so we have now some maps of clay mineralogy for the surface and subsurface all across Australia. And as you can see, if you know anything about the geography of Australia and the soils, you can see that, you know, the mapping and the method picked out quite well all the smectites in the Channel Country and in northwestern New South Wales, and it picked out which are highly smectitic, of course, or the kaolinitic upper regions in the centre of Australia, Western Australia, and so on. These images are just composites of the three maps. So we have a map of kaolinite, illite, and smectite, but I thought it was a nice way to put them together so that you can see that the red bits are kaolinitic, the blue bits are smectitic, and the green bits are illitic. Now, similarly, we measured those samples, and then we looked at different parts of the spectrum. So remember that by measuring the decibel range, we have bands that represent, or wavelength regions that represent the green, red, green, and blue. So we integrated those bands to derive a red, green, and blue, and then we mapped those using some geostatistical methods. And I put this little map of Australia on the left there, which is a remote sensing image by Landsat. And you can see the correspondence between the measurements, and they should be the same because it's the same, you know, Landsat measures red, green, and blue, and that's what we did. We just did it slightly different. The advantage of our map, of course, is that it represents the surface, so it's not affected by vegetation, which is kind of useful for lots of different types of modeling. Because the spectra, as you might remember, also measures hematite and goethite, so we also mapped the probability that a soil has hematite or goethite, and that's the map in the middle. And finally, one of my interests a little while back was to see whether the spectra could tell you about soil type, because if you remember, I said that soil measures, or the spectra measures soil composition, all the things that make up soil. So what we did then is we summarized all of the wetlands in the near-infrared spectrum using principal components, and then mapped the first 3 components. And similarly here, I overlaid those in a color composite, so that the blue regions are the kaolinitic soils, the green regions are the smectitic soils, and the red regions are soils that have more organic matter, so the things that the spectra really measure. So my idea was then to try and derive a more objective soil classification, but I never got that far. As I mentioned, organic matter, organic carbon is more difficult to measure with the spectrum, more difficult to pinpoint the exact absorptions that relate to specific organic materials. So we need to do some modeling. So again, this is a few years back as well. We have funding from the Department of the Environment, the people that run the national inventory, and they wanted us to develop a soil carbon map so that they could use this in the national greenhouse gas inventory. So we did that, but we couldn't have done it without spectroscopy. So we had data from the SCAP project, which is a soil carbon project that was running in 2013, I think, around then, but it covered only agricultural regions. So these are the green dots. We had some legacy data from the soil archive, but it was rather incomplete, and those are the black dots, right? We wanted this baseline to represent a particular time period, and that's why there's so few of those black points, because the archive has lots of samples, but we wanted samples that represent only a particular decade. So what we did is we used spectroscopy, the spectral library, to model carbon and then estimate on all those red dots. Right? And so then we had estimates of carbon across the country, we harmonized them, we did lots of things to those, and then we modeled it spatially, and we derived the baseline, carbon baseline in 2013 or 14, which told us how much carbon there is in Australia and in various systems. More recently, we've been working with a number of colleagues from different organizations because we want to update that baseline. So that baseline was really agricultural, and there was a lot of samples from other systems, but very few from forests. And definitely that didn't cover any of the coastal marine systems. So we wanted to derive a baseline that will better represent forests and also cover the blue carbon regions. And so we've been developing that using very similar methodologies because we wanted the baselines to be comparable, and we've got some results. So you can see that the blue areas are also fairly well estimated. Terrestrial systems have less carbon on average than the blue systems, which makes sense. But as a whole, if you look at the totals, the total stocks of the blue carbon systems are much, much smaller. And that makes sense, of course, because they occupy such small areas. So we're analyzing this data, and Lewis, who's an ex-Murdoch student, is writing this up at the moment. We looked at carbon composition. Again, this was follow-up study funded by the Department of the Environment. And it was really just to parameterize the full carbon or the Roth-C component to better estimate our emissions. So they wanted, because they're using Roth-C, Roth-C uses measures of carbon composition, so the mineral associated with the humic carbon and the resistant. And so we did a similar thing. All the gray dots there are samples where we had no carbon fractions. The red dots are a library that was collected through the SCART project and a subsequent Within the Research Gap 2 project. Those models were developed and then we predicted, and all that's been published, of course, by Jeff, myself, and others. And we were able to predict the fractions. I won't tell you too much. I think a lot of you have heard about this before already, so I won't tell you more about that. Another example that, you know, in 2013, we had the Soil and Landscape Grid of Australia project, a CSIRO project, a CSIRO-led project that involved all the states and territories in Australia. And the idea was to map soil properties so that Australia would then have a complete set of soil properties across the country that people could use for lots of different purposes. Again, you can see the the black and the green dots is where we had actual data. With only those data, our maps would have been highly uncertain, right? So what we did is we complemented the real measurements with estimates of those soil properties using spectral estimates, which are the red dots, and then we could actually do the mapping of the properties. So we mapped 13 properties in total in 3 dimensions. So I should have said that the idea here was to map soils at different layers, so the 0 to 5 layer, 5 to 15, and so on, all the way down to 2 meters. So that the spatial modeling had to be done in 3 dimensions. And we produced that. That was supposed to be a dimension that's not working, but okay. That's applications of spectroscopy in digital soil mapping. And I've shown a lot of continental scale stuff, but that doesn't mean that you can only do continental scale stuff with this technique. There's lots of examples that I could have shown you where we're doing things at regional scales or field scales. Okay, just a couple more slides just to show you how spectroscopy and sensing are helping us better parameterize our carbon models. So we worked quite a lot with the Emissions Reduction Fund to look at developmental methodologies for soil carbon assessment. And so we looked at how we could use a model to derive estimates of carbon change over time. So this doesn't mean that we want to replace the measurement with the models. This is just an example showing how models could be used in one of those systems. Measurements are essential to modeling, right? I don't want to say that you can replace measurement with modeling. So in fact, we use spectroscopy and sensing to derive the parameters that you need to input into these models. So you use spectroscopy to measure things like carbon content, particular carbon, mineral-associated carbon. You measure sensors like gamma attenuation to measure bulk density, and so on, and then you can use those in the model. And what we found is that the models can work more accurately if you do have sensor data, even though it might be a little bit less precise than using very, very precise few measurements that are made, very precisely. Yeah. Jumping scales again. Over the past couple of years, we've been developing a framework for consistent soil carbon simulations at different scales. And the idea here is that we want to build this. We wanted to build this framework because I think the model can be used at whatever scale is most appropriate. So if you're interested in continental scale simulations, okay, well, if you have a consistent framework where you do the right things to the data, then you can use that model to answer those questions at that scale. If you need it at a farm scale, okay, well, then you have a consistent approach for actually making those simulations. So this framework is kind of unique and different to what other people do, because what we do here is we initialize the model with sensor-based measurements of the carbon fractions, which means that we don't have to run these very long spin-up simulations to get the model running. So we initialize them with actual data. And as I said, the simulations are consistent, and then they can help us to answer different questions. And this is ongoing work, so I haven't really put much here. but we are running the model into the future to answer questions like how will future climate affect soil carbons in Australian managed and unmanaged systems? How can management practices mitigate any of the possible loss? And to answer the million-dollar question, which bits can sequester carbon? and so this is really ongoing work. Okay, some summary and recommendations. I know I'm a little bit over time, but I'm almost there. So spectroscopy, different methods, different advantages and disadvantages. Spectral range depends on application, the instruments that you can afford, the costs and so on. The spectra encode information on organic minerals. It can be used as a fingerprint for soils. You might have heard that before. Standardization is really important, but and there are people working on proper standards for spectroscopy. But really, I think some of those standards tend to use unique standards or unique materials for standardization. Really just complicate what is really a very robust and simple approach. So more effort should be placed on the standardizing of analytical methods rather than spectroscopy, I think. Now, the modeling needs to be done with understanding of soil science, right? And soil spectroscopy. It's not a data science exercise. As I said before, don't use a sledgehammer to crack a nut. If you can do things simply with a robust statistical method, do it. Don't go for the machine learning, which is also much more difficult to interpret, of course. Don't expect miracles. There are some cases where spectroscopy is not going to work for whatever reason, right? And I've listed some of those there. Just remember that if you're using it, follow best practice. Remember that GIGO principle. Don't lose sight that there are other methods. Spectroscopy is really good, and I really like it, and so should people. But it's not going to do everything. Not one single technique is going to do everything. There are lots of other things. But the whole idea, I think, is to, if I was developing a system, is to try and find the sensors that you can use complementarily, so that then you can measure the whole sort of system rather than trying to measure something that's not possible with just one technique. Some final remarks. Like most methods, soil spectroscopy has had its hiccups. I think that practitioners have been a little bit over-optimistic and oversold the technique. But as it develops, I think that we understand those methods better. And so we are learning how to use them better in practice and in research. So we're becoming a little bit more adept at using these methods properly. As I said to you before, we don't want to replace conventional methods, at least not entirely. I think for some things you can. So those mineralogical assessments, I think we can do that directly with the spectrometers. Actually, we don't need to do X-ray. But for estimating soil properties, functional soil properties, no, we don't want to replace conventional methods. We've got to work alongside conventional methods to be able to get those. The advantage, of course, is that if you use them complementarily, the total cost of your analytical thing can significantly. That's what we want. It's become an essential tool for measuring and monitoring soil. And we need that because more and more we need more and more information to actually make those decisions that we need to make to make the world a safer, better place, I guess. So I think with that, that's my final. I really want to thank my little group at Curtin and my many collaborators that have helped me to produce those nice graphs. My little group is starting to produce some nice papers and some nice results, so I would really like to thank them. And before I go, if you're interested, go and have a look at this opinion piece of soil spectroscopy. So we wrote this and it's just been accepted. It's just kind of cool. Took a week and a half to get accepted. My fastest paper ever. But the thing is, right, this guy, this Professor McBride from Cornell, I think, wrote this opinion piece on soil spectroscopy with a fairly outdated and pessimistic view of the technology. So a group of us got together and decided to write a sort of critique of his opinion, write our own opinion. And I would encourage you to go and read it if you're interested in the source. Thanks.
Thanks for that, Raphael. And there's nothing better than a bit of debate in science to help with publication counts. So absolutely sure to open it up. So we'll open up the floor to questions. Any?
How far away are the commercial labs that have the MIR away from having it as a commercial service? Do you know?
I think that in terms of the analytical method, that having the spectrometers and some protocols to actually measure the spectrum, I think they've been there for quite some time. The problem is that localization issue, right? So it's not a matter of just building a big library and then building a model and then predicting, because we know from a hell of a lot of experience. I mean, Sarah tried that in the 90s, I think, that it doesn't work. So until we develop these algorithms that can mine the information from those global libraries and do something clever so that then you can estimate unbiasedly at a site, yeah, we're not there. Those methods are starting to become more and more available. One thing that I was going to show today, and I did this at the time, is that we built a software tool that implements our methods for localization. I didn't show it today because, well, we haven't finished it. I thought we were going to have it finished, but it's going to be available for free. Right? And I think these things, it's this type of thing that we need to make spectroscopy more widely adopted by people. And this is the sort of thing that labs need, I think, for it to be more widely used.
Other questions?
Hi, Raphael. Very interesting topic. Because I have a background in remote sensing, and I mostly use the satellite remote sensing to monitor, have research on the vegetation on top of the soil.
Right.
Yes, I'm quite interested in the continental soil mapping. Do you think how much accuracy can the Landsat estimate the soil property, like carbon? Because they are like the visible and the reflectance is a lot of noise, like obtained from the satellite image.
Absolutely. Remote sensing by itself can tell you about soil carbon. Right? It might tell you about soil carbon in the bare bits, but in the really top little bit of the soil, a few millimeters, if that. Right? But you know, for all of that digital soil mapping, we did use Landsat because it's a good predictor. Right? But the Landsat images, like most other images, need to be processed quite a lot, and you need to do some temporal analysis to try and find the bare areas at different times. But once you do all that analysis and you can get a relatively bare soil layer, then you can use that as a predictor or a covariate in your models to estimate carbon. And it's not the only one. You use things like climate and Landsat and other things, of course, but I don't think that these remote sensing images can be used by themselves to give you an accurate estimate of carbon or any other soil property to a reasonable depth.
Yeah. Question up the back there, yeah. And then Fran.
Rachel.
Thanks, Raphael. That was great. As you probably guessed, my interest is in restoring soils from a degraded state. Do you think this is the tool for the job, or should we jump to the sort of biological properties of soils that are going to be changing through time?
Yeah, I've been trying a little bit to look at how, if these technologies can help you quantify biology. I haven't shown it here, but it sort of can, but not directly. It's a very indirect way to sort of assess biology. What we've tried is to combine spectra with other data, which we thought would be useful. So things like, you know, some sort of landscape attributes, more water, climate data to try and see if they could help us to better explain or understand the distribution of these microbes. But it's kind of tricky. So what I actually think is that spectroscopy is probably not really the tool for it. What is the right tool for it? I'm not quite sure. And it also depends what you want to do. So one thing I will say is that if you're interested in getting some broad assessment of, microbial diversity, fungal diversity, or bacterial diversity, you could use spectroscopy like I described, not by itself, but using it in combination with that data, and potentially also with some meta barcoding to put it all together to give you a, reasonable picture of the distribution of these microbes and their diversity. However, if you're trying to assess a mine site and trying to use it to see whether that soil is alive again, I'm not sure. I think we need something else.
So mine wasn't actually a question, it was kind of an offer, I guess. Raphael, when you finished your software development, maybe we could host a workshop or something for people working with IR data.
Yeah,
that would
absolutely.
help them.
That would be fine.
Yeah, and then we just do a tutorial type workshop or something like that.
Yeah.
Rachel, it's not along the same lines, but I remember Dan, and you might have more comment. Years ago, when we were playing with MIR and paddock data, I was on a much smaller scale. Quite a lot of success getting something like potentially mineralisable nitrogen, which was an indicator of, I guess, biological activity. So I know you didn't do lots of different parameters, but I guess I was curious about your.
I think it's as Raphael said, all the biological measurements, when you look at them, are correlating a lot with soil attributes. So chemistry and mineralogy and organic matter, which infrared technology is very, very accurate at, helps describe the habitat, the environment the soil beta are living in, so you get that co-correlation.
So it's understanding those embedded sort of mechanistic controls and then using the IR to predict potentially.
Yeah.
I don't know.
Yeah.
When you had the whole Australia map and then you said you estimated for getting the soil types, how did you do that?
The soil types. I looked at the spectra, those 5,000 points. Then I summarized them using principal components. So and then I took the first three components that explained, 80% of the variance in all of that data. And then I mapped those components. And then when you look at the loadings of those components, the first loading was really mineralogical, particularly kaolinitic, if I remember right. The second one was also mineralogical, but smectitic. So these have different absorptions in different parts of the spectrum. And the third one was really broadly organic. So when we mapped them and put them together, pictures started to emerge and so, oh wow, that's really mapping soil type really. In subsequent work, we also mapped the Australian soil classes with the spectrum. And we could do that very accurately. Okay? The advantage of doing it with the spectra is that we also have estimates of uncertainty for each class. So we could actually say, we are really sure that this soil is a bird soil, or perhaps it could be, a soda soil a little bit, and we could quantify that. But the spectral method combined with this machine learning was actually rather useful for mapping soil type.
And as you went deeper, with some soils were you quite confident still?
Yeah, most soils were mapped fairly confidently. There were some, so when we compared it to the traditional Australian soil classification map, they broadly overlapped, of course, but the boundaries were a little bit different in some places. My whole aim with all of this was to try and see whether we could get rid of the AAC altogether, actually, and derive some sort of quantitative way, so that then, you're not relying on saying that this is a soda soil based on somebody being in a pit and tasting the soil. And so you can take the spectrometer and put it down and get a spectrum and say, let's classify that. Never got that far.
So my last question.
I'm just wondering if combining NIR with MIR, does it always give better result or it depends on the soil type?
I think it's wasteful because if you remember my description of this, these spectral ranges, actually what information is in the mid-infrared is the same that's in the near-infrared, only weaker. So when you combine them, you get very little benefit because you've already got that information in the MIR. What I find useful is combining it with the visible, because the visible has unique information that one of the other spectral lenses don't have. But again, you have to be a little bit careful going into the literature because you will find lots of papers that people say, oh yeah, we combined the spectral ranges and we got better prediction. And then you think about it and you go, why did you get this better prediction? How? And then you realize that yeah, it's some crazy modeling thing that they did, and for that, whatever reason, they got better results, but it doesn't make sense.